Okay, so today we are going to be talking about DNA slash RNA. Okay, so first off, we need to know that there are two types of nucleic acids. So remember, there's four types of organic compounds, which are compounds that include carbon, and those are carbohydrates, nucleic acids, proteins, and lipids. And when we are looking at nucleic acids, we have two types, and those are going to be DNA and RNA. So DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. And they're the hereditary material in our body. So um, right here we see a couple differences between DNA and RNA. So DNA, obviously the name, that comes from the sugar that's present in DNA and RNA. So DNA uses the sugar deoxyribose, while RNA uses the sugar ribose. DNA is double-stranded, and this is where it gets its double helix shape from, and RNA is single-stranded. DNA uses the nucleotide thymine, while RNA replaces it with uracil. So now we're going to look at the composition of DNA. So DNA is made up of nucleotides, and these are made up of one phosphate, a 5-carbon sugar, and a nitrogen base. So the 5-carbon sugar is what we were talking about before, and for DNA that's going to be deoxyribose, and for RNA it's going to be ribose. So we have 4 nitrogen bases for DNA, and they are adenine, guanine, thymine, and cytosine. Um, uh, this is where we see that uh, there's a difference in RNA and DNA, and this is because RNA uses uracil instead of thymine, and when we are looking at the bases more carefully, we're going to see that adenine and guanine are purines, and thymine and cytosine are pyrimidines. So purines are made up of 2 carbon-nitrogen bases, so it's going to have 2 rings, and thymine and cytosine are made up of 1 carbon-nitrogen ring bases. So that's basically the difference between them. Also, purines are generally larger, and pyrimidines are smaller. So we have the discovery of DNA, and these are just some scientists that you should know. So Frederick Griffith, um, he did the so he discovered that genetic information can be transferred from one cell to another, and he worked with the mice and the strains of bacteria. Rosalind Franklin and Maurice Wilkins used X-ray crystallographic imaging, which helped with the development of the double helix, and we can see a picture of it in the top right corner. That's what they were looking at. That's how they discovered the shape of the double helix for the DNA. James Watson and Francis Crick developed the double helix model of DNA, although many say they should not have deserved the credit because most of it came from Rosalind Franklin, but they did win Nobel Prizes for their contribution. Erwin Chargaff, he discovered base pairing rules. So he discovered that adenine pairs with thymine and guanine pairs with cytosine. And then Matthew Stanley Meselson and Franklin William Stahl discovered that DNA replicates in a semi-conservative way. So when we're talking about DNA replication, we know that half of the strand is the old while half is new as the new bases are added. So that's called the semi-conservative way of replicating, and he was credited for discovering that. So this is more about DNA replication. So first, DNA helicase, which is an enzyme, begins the process by unzipping the double helix, and then DNA polymerase goes down and it adds nucleotides to the strand. So it's adding new nucleotides to the old nucleotides. So that's the semi-conservative method. And DNA ligase connects the two strands of DNA. So think of it like a zipper. The DNA helicase unzips it, and then the DNA ligase connects it back. So now just some review. We take about 30 seconds to answer these questions. Okay, so what does adenine pair with in DNA? So remember, adenine is going to pair with thymine, and then guanine pairs with cytosine. What does DNA polymerase do? So DNA polymerase goes in after the DNA helicase, and it's going to add the new nucleotides during DNA replication. And then what is mRNA and what does it do? So mRNA is a messenger RNA, and it carries genetic information from the nucleus to the ribosome, which is going to be crucial for DNA replication.